Okay. Hello everyone and welcome back to our COVID webinar series looking at the role of proteomics. Today we're joined by two speakers, Dr. Jeroen Demmers and Professor Andrea Pichlmair. Just a few housekeeping things before we get going. As always, we're using our Slack channel for questions and discussion, so please join us there to ask questions, share any thoughts and discuss the work. Slack helps us to prioritize the questions that others want to hear the answers to, so please use the thumbs up to let us know those questions you'd like to hear answered. Slack also allows the speakers to answer any other questions or follow up after the talks is finished. And for those needing an attendance certificate for this webinar, the details will be available on how to get this after the last slide. Once again, we'd like to say a big thank you to the European Proteomics Association, the British Society for Proteome Research, the Young Proteomics Investigators Club, and the London Proteomics Discussion Group for their help and support in setting up this webinar. Thanks also to the London Biological Mass Spectrometry Discussion Group, the London Metabolomics Network, and the News in Proteomes Research blog for promoting this event. And we're also grateful for Imperial College London for providing us with webinar support. And a huge thank you to both of our speakers for their time today. We're also pleased to announce that our next webinar will be on Friday the 7th of August at 2 p.m. British Summer Time for our second in the new series of non-COVID webinars, where we're pleased to announce a guest chair, Catherine Lilly, who will be hosting our speakers, Dr. Ben Neely from NIST in the US and Professor Claire Ayres from Liverpool University here in the UK. So I'll introduce you to the first of today's speakers. We'll be hearing from Dr. Jeroen Demmers. Dr. Demmers is Director of the Proteomics Facility at the Erasmus University Medical Center in Rotterdam, having completed his PhD in the Heck and Killian Labs at Utrecht University and a postdoc at the Rockefeller University in New York. His research interests in mass spectrometry and proteomics extend to quantification, targeted proteomics, PTM analysis, transcription, gene regulation in health and disease, and chromatin biology. Today, he will give a short talk about targeted proteomics for the detection of SARS-CoV-2 proteins. So it's over to you, Jeroen.
Great, thanks, Joe, for this nice introduction, and thanks for having me. I'll put up the title slide. So this talk will be about the targeted proteomics for the detection of SARS-CoV-2 proteins, work that we did over the past few months. So the causative agent for COVID-19 is SARS-CoV-2, and I guess you're all familiar with the numbers that I'm showing you on the next slide. Yeah, here. I'm sure you're all familiar with these numbers over here. Obviously, extensive testing for SARS-CoV-2 is crucial to define sites of outbreaks and therefore to take measures to contain and stop the spread of the virus. So there is a need for rapid, sensitive and specific diagnostic tests, and the golden standard of SARS-CoV-2 tests are PCR based. Still, the coronavirus study group has urged people to take a proactive approach to develop alternative and additional diagnostic tools. Not only because the insufficient testing capacity, but also for research purposes, of course. So as a proteomics lab, we asked ourselves whether mass spectrometry could be used as a tool for testing. And in this talk, I will summarize what we've done so far. I apologize for the confusion. The slide, the PowerPoint presentation seems not to work smoothly. Dan, could you please help me? I want to go back to the previous slide. Yes, thanks. So the SARS-CoV-2 virus is built up of an RNA strand, several different proteins, spike envelope, membrane, and nucleocapsid, and is surrounded by a lipid membrane. The precursor protein ORF1AB is translated and then proteolytically cleaved into functional proteins. And one of them is NSP1, which was actually recently shown to be involved in the almost complete shutdown of the host protein translation machinery. This cartoon is perhaps more realistic in that it shows the protein abundance ratios better. Nucleocapsid is by far the most abundant protein in the virus particle. And we confirmed this ourselves also using label-free quantitation mass spectrometry. And I'll come back to this in a minute. We set off by defining the target tryptic peptides that have to serve later on as the target peptides in our targeted proteomics assays. And for this, we took Vero E6 cells infected with SARS-CoV-2. The virus was inactivated by boiling the cells for 20 minutes in a Lemley buffer containing a lot of SDS. And this is our so-called test sample. The rest of the protocol is pretty straightforward. Proteins were precipitated in and dissolved in urea before being digested. We were quite lucky that just before our institute was shut down, the brand new Orbitrap Eclipse was installed, and literally the first proteins measured on this beast were SARS-CoV-2 proteins. So for most of the other samples that I will describe later on, difficult matrix samples, patient samples, we also used the single pot SP3 method that was developed in the Krijgsveld lab, optimized for small sample amounts, and this works actually very well in our hands as well. So this is just an overview of the results of this test sample. We could identify all SARS-CoV-2 proteins with decent coverage percentages. And apparently there's a lot of virus in these cell cultures. So the cells inside the cells and secreted from the cells. And I should emphasize here that we do not exactly know how much of these proteins are really assembled in active or infectious virus particles. Based on the MaxQuant label-free quantitation data, we think that almost 90% of the total virus proteome is actually composed of nucleocapsid protein. So these are just a few fragmentation spectra, and these MS fingerprints were used to design the PRM workflow that we used. And for those of you not familiar with PRM, so in PRM, we let the mass spectrometer focus on only those targets that we are interested in. And this makes it possible to lower the detection levels. Next, we measured a dilution series of the cells plus virus test sample in a targeted PRM assay, where we focused on a few highly responsive nucleocapsid and spike peptides, and these are just two examples over here. For the lower limit, we could go down to about 20 picograms of total protein amount, and this then would correspond to slightly less than 1 picogram of nucleocapsid protein. And these are actually the same results in a slightly different format. And you can nicely see the linear relationship between the input and signal over several orders of magnitude in this PRM assay. We then did some very basic calculations to find out which number of viral particles this would actually correspond to. Important to keep in mind here is that the lower limit of 20 picograms is of total protein, so Vero E6 proteome and virus, and that in the dilution series, the matrix, so the cell proteome, was also diluted. I make a few assumptions here. So the composition of the of an RNA-based virus is about 10% RNA, 70% protein, 20% lipid. Another assumption is that 90% of the SARS-CoV-2 proteome is nucleocapsid. So that would then be about slightly over 1,000 copies per particle. And this number corresponds to, or at least is in the same ballpark as copy numbers that were published in the literature for other RNA-based viruses. And so if you take these assumptions, the lower detection limit would be about 10,000 virus particles. And there are clearly a few very big ifs here, and this should be considered no more than just a ballpark figure. So next we did some analyses with samples that we think are more similar in overall composition to patient samples. And these are actually dilutions of virus stocks into secretions of organoid-derived human airway culture cells. And in fact, these secretions can be regarded as a proxy or a substitute for mucus. These secretions consist mainly of proteins such as uteroglobin, lipocalin, and so on. So in this case, we made dilutions in a constant background of mucus and started doing some PRM assays. And the results are shown over here. So for this target peptide, a nucleocapsid peptide, we could still see a decent signal using PRM after 100-fold dilutions of virus stock into this so-called mucus. And we know that PCR was able to detect virus levels at higher dilutions. That shouldn't be surprising. The problem here is that we do not exactly know how much virus is in the stock, because we don't know how the so-called TCID50 corresponds to the number of particles. So this TCID50, the 50% tissue culture infective dose, is the measure of infectious virus titer, which virologists use to determine the amount of active virus. But it's not entirely clear how this value corresponds to numbers of particles. So we don't know exactly how many virus particles are present in these dilutions. So after these preparative experiments, we took this further to patient samples and asked ourselves whether we could measure viral proteins in them. And this whole endeavor started off in a quite disappointing manner, I would say, because we weren't able to detect any viral proteins in material from swabs and nasal and throat rinse solutions. And we think the main problem here is that diagnostic departments deliver all patient material in UTM, so-called universal transport medium, and this contains usually a lot of serum and or albumin, according to their standard protocols. But for proteomics, of course, this increases the background to a huge extent, and that's not what we want. But then sometime later, we could get some sputum samples, and we had already seen in the literature that sputum and also saliva are maybe even better sources of material for virus detection. For these samples, I don't know exactly how they do it, but I think the patient just spits on a piece of glass. And the material is then fixed with 80% acetone, and that's it. And then we applied either direct digestion on the glass slide, or we used sp3 again. And the results here actually look very promising. So we targeted several nucleocapsid and spike peptides. And this here is just an example of one such target peptide. And using PRM, we could actually nicely detect the target peptide in these glass slide samples, as you can see here. So the fragmentation patterns here are virtually identical to those of the positive controls. And the positive controls in this case is the pure virus stock. And if you compare this to the results of the swabs on the next slide here. So these are the results of the swabs, and these are the results of the glass slides. It is clear that the sputum glass slides work way better in our hands. And this is true not only for this target peptide, but also for several other target peptides, including those for spike protein. So we then incorporated the use of heavy stable isotopes as spike-ins in the sample for two reasons. So first, it's possible to more accurately quantify target peptides this way. And second, there is this protocol called SureQuant in the Thermo software that makes it possible to let the heavy labeled peptide act as a trigger for PRM on a target peptide in order to gain some sensitivity. So we applied this method. This is what we did for, in this case, the same target peptide as I showed you before. These are the chromatograms of the heavy peptides, and these are the ones for the target peptides, with clear signals for the sputum samples and less to no signals for the swab samples, as you can see in the lower part of the figure here. So this slide basically shows you that the increase in sensitivity, if you apply aqua peptides in combination with this SureQuant method, is about 2 times and expected to be somewhat higher after some more optimization. So these are all preliminary results, obviously. And also what you can see here is that we can observe some additional transitions when we use this protocol. Right now we are expanding our sample cohort and try to collect more sputum glass slides. So most of the mass-spec work was done by Karel Bestarosti in my lab, and most of the virus work was done by Mart Lames in the lab of Bart Haagmans here at Erasmus MC. Part of the work, excluding the patient data, was posted on bioRxiv some time ago, and I hope that the complete manuscript will be published somewhere soon. And I thank you for your attention.
Great, thanks, Jeroen. That was really interesting. We have a couple of questions already coming in on the Slack and other places. So the first of these questions is, are all the peptides that you used in the PRM assay, are they proteotypic and specific for SARS-CoV-2? And I think this question is in the context of other similar coronaviruses. Have you checked that they are really specific for SARS-CoV-2?
Yeah. So we actually, when we selected peptides, we chose for the highest intensity ones, of course, but also for peptides unique to SARS-CoV-2. So some of them are unique for SARS-CoV-2, some of them are unique for either SARS-CoV or SARS-CoV-2.
Great. And then another question that's actually come in. So did you consider any PTMs when you were optimizing this? How many transitions were you considering for your PRM design, and how many optimization steps did you do before you reached the assay that you ended up with?
No, we didn't include any PTMs, was the question, right?
Yeah.
No, we didn't, not yet at least. So these are all very fresh results, I would say. We have at least 5 or 6 transitions per target peptide. That's basically our standard always for PRM, and this is also true in this case. And I think the aqua peptides, the use of aqua peptides is the best control you can get. And if the transition patterns are similar to your aqua peptides, then I think you're in.
Great. Yeah, I think that's it for the questions that have come through so far. There may be some more at the end, but we'll move on now. So I'm going to introduce you to the second speaker from today, and that is Professor Andrea Pichlmair from the Technical University in Munich in Germany. Andreas obtained his PhD from the London Research Institute for Cancer Research in the UK, and then moved on to his postdoctoral work at the Centre for Molecular Medicine at the Austrian Academy of Sciences. He set up his own lab at the Max Planck Institute of Biochemistry in Munich in 2011, and his research focuses on the interaction between viral pathogens and their host organisms. He primarily uses mass spectrometry and similar large-scale techniques in order to conduct system analyses. Today, he's going to be speaking to us about how multi-level proteomics reveals host perturbation strategies for SARS-CoV-2 and SARS-CoV viruses. So it's over to you, Andreas, when you want to take control.
All right, I want to thank you very much for the invitation. It is a really nice webinar. Right. So we just try to change slides here. Okay, so the main interest in my lab is actually the interaction of viruses with the host, particularly focusing on how the cell would prevent virus replication in the cell, and also how the innate immune system is equipped to defend these viruses, because this innate immune system is very universal for many different pathogens, but yet it is also very specific to target these pathogens in particular. We are studying these interactions using mostly proteomics and other omics approaches. And we ask basically which cellular proteins are, for instance, binding to viral proteins or to viral nucleic acids. And we try to deduce mechanisms as how these different entities are interacting with each other. The long-term aim of this approach is that we try to identify antiviral treatment options that can be used for individual viruses or that can be acting pan-viral. And as Jeroen just introduced, this was fine until very recently, until a new pathogen came out, which is SARS-CoV-2, and it was basically suddenly everywhere. And of course, we got very interested in the interaction of this pathogen with the cellular proteome. If you consider now this virus as a pathogen, of course, you may think about interests of the pathogen itself. The virus wants to replicate. It acquires cellular resources, and on the other side, you have an interest of the host to defend the pathogen and to overcome this virus infection. There is a variety of different questions, of course, that one would ask when getting in contact with such a pathogen. One is, for instance, what are the interactions between this new coronavirus and the host proteome on a molecular level? Why is this different to other coronaviruses, such as the 2003 outbreak coronavirus? And which pathways are linking the infection events to pathology. And omics approaches are, of course, very useful for this to study. And I want to mention just a few of really stellar publications that came out recently on different aspects of how this virus is activating or interacting with the cell. For instance, Neven Kroven published this protein interaction map of viruses or viral proteins with cellular proteins. There's also data on proteomics changes after virus infections, and you have seen Christian Lynch here on this channel too. There's also phosphorylation and phosphoproteomics data also from Neven's lab, and also some about clinical proteomics and transcriptomics data. And I want to apologize for everybody who I cannot list on this slide. There's an amazing effort of many people that help us to understand this system. One drawback of this individual omics approaches, however, is that they very often use very different systems to study this virus. And therefore, the comparability and the intersection between these different omics approaches is sometimes a bit difficult. And to tackle this, we basically set out to establish a system that allows us to test multiple omics approaches in one single cell type, so that we can compare these different layers of interactions, and that we inform one omics approach with information from another. And here we are mostly focused on a cell line called A549, which is a lung-derived cell line. And we studied this cell line in the context of single protein activities, whereby we test, for instance, the interactions in the proteins with the cellular protein in the cells. We test for the changes on the virus infection context on a quantitative level, looking at proteomes and transcriptomes. And we're also testing post-translational phosphor modifications in this cell line after SARS-CoV-2 infection. And in order to just guide you through this, we are basically studying this coronavirus expression in these cells. So we are expressing these proteins in the stable versions in these A509 cells with an HA tag. We precipitate these proteins and we analyze associating proteins with using mass spectrometry. And we did that for both the SARS-coronavirus 2 and the 2003 outbreak of SARS-CoV, as well as a few members of human coronaviruses. And this allowed us to generate a joint interaction map of SARS-CoV-2 proteins and SARS-CoV proteins with the cellular proteins that you can see here as gray nodes. And altogether, we identified more than 1,000 interactions between these viral proteins and the cellular proteins. Many of these interactions between these coronaviruses were shared. So we find the majority of these proteins being shared between SARS-CoV and SARS-CoV-2. But there's also some unique interactions of SARS-CoV-2 or of SARS-coronavirus. And you can see these interactions here labeled in orange if it's a SARS-CoV-2 specific interaction, or labeled in red if it's a SARS-coronavirus interaction. And they can now look into this in more detail, and by considering the activity of these associated proteins. For instance, and there are some quite interesting targets. For instance, the ORF7A protein targeting proteins of the nuclear export complex that is important for export of RNA from the nucleus. There's also some specific activities of certain ORFs, such as, for instance, the ORF3A finding to the MHC complex proteins. And very recently, there was a paper appearing in bioarchives suggesting that this is a way that the virus can, for instance, perturb presentation of viral proteins on the surface and thereby limit the activity of the adaptive immune system. Also, some quite specific interaction we identified is between the ORF8 protein and the members of the TGF-beta complex. And one should note that the TGF-beta complex itself has previously been linked to pathology of influenza viruses and other respiratory viruses. This seems to get out of place after virus infections, and this can be positively linked to pathology of these viruses. So overall, we got a very nice interaction network that tells us about the interactions between the virus proteins and the cellular proteins. And on top of this interaction map, we also mapped the activity of these individual proteins on the protein expression in cells. So we basically measured protein expression in cells that stably express these proteins using a total proteomics approach. This basically allowed us to deduce the activity of a single viral protein in this experimental system, and we could thereby identify changed proteins, changed on the basis of their abundance in these transduced cell lines. Some of these proteins also enriched for GO terms. One example here, for instance, is the ORF9b protein of both the SARS-coronavirus 1 and the SARS-coronavirus 2, which appears to be quite dramatically affecting mitochondrial activities. And this could be through interactions with proteins such as the TOM70 protein, which has previously been linked to mitophagy. So we could assume that the activity of this protein may induce mitophagy, and that would relay to a dramatic impairment of mitochondrial activity. Besides the single proteins and the activity of the single proteins, we also considered the antiviral particle. And for that, we basically generated cells that are expressing ACE2, which is the SARS-CoV-2 receptor on the surface. And upon expression of this SARS-CoV-2 receptor, we can see an infection of these proteins after exposure of the virus, of course, while the parental cell line seems not to be so infectable. And we now use this cell line to test protein and transcript expression in a time-resolved manner. And we could see several hundred transcripts, for instance, being dysregulated after virus infections. as well as several hundred proteins that are changing their expression patterns over time. What we can see in general, and what is in line with other people's reports, is that the proteome changes after infection with this virus do not seem to be as dramatic as changes that we can see on the transcript level. But yet this transcript level tells us about regulations that are happening in the cell after virus infections. And there's just one example I want to show here, which is that these cells that are fully infected with SARS-coronavirus 2 would only induce a relatively minor innate immune response, as measured by, for instance, transcript levels for interferon beta, which is one of the main antiviral cytokines that are elicited by viruses. And this very minor induction corresponds to a quite low induction of antiviral proteins, such as, for instance, the MX1 protein, which is an interferon-stimulated gene. But in contrast to this interferon-regulated class of proteins, we can see activation of NF-κB-related genes, such as, for instance, IL-6 and CXCL2, which are pro-inflammatory cytokines, and that contribute to a pro-inflammatory state of, for instance, an infected lung. And again, this pro-inflammatory state had been linked to pathology of viruses in respiratory diseases. Besides this proteome and transcriptome level, we also tested for differential phosphorylation after virus infection, if it counts. Oh, here it is. And we identified about 1,500 phosphosites that changed dynamically after virus infections. And we measured this in two different modes, in DIA and data-independent acquisition and data-dependent acquisition. And what we learned from that is that this virus already at early times after infection, for instance, changes certain pathways in the cell that are related to DNA damage response and to ERK signaling. And considering the entire time frame of these infection events, we can also see MAP kinase activation, chemokine signaling being perturbed, DNA damage and regulation of transcription. And altogether, we could map motifs that were differentially regulated in the context of virus infection. And we could identify around 160 kinases that are differentially active in these virus-infected cells, suggesting that there's a quite dramatic effect of PTM activities in these virus-infected cells. Besides phosphorylation, we also looked at the ubiquitination of proteins, and we could also find some relation between the phosphorylated proteins and ubiquitinated proteins, which basically is in line of accumulating post-translational modifications on individual proteins. And we could, for instance, identify 118 cellular proteins that are changing both phosphosites and ubiquitination sites in an infection context. We could not only look at cellular proteins, but also viral proteins. And here we, for instance, have evidence of phosphorylation events that are happening on viral proteins. For instance, here on the N protein, where we could also map a number of kinases that we suggest to be active on this protein to phosphorylate these residues. Besides N, there's also SM and ORF9P, and there's also quite nice data on the phosphoproteomics data set from Neven Kroven on particularly this case. Besides phosphorylation, we also find ubiquitination that is quite dramatically affecting viral proteins. There's, for instance, ubiquitination sites in ORF3 in the N-terminus. And besides these ubiquitination sites, we could also identify motifs for TRIM47, and indeed ORF3 was binding to TRIM47 in this interaction data set that we identified, suggesting that there may be a functional link between the recruitment of this E3 ligase to this ubiquitination event in the N-terminus of ORF3. And besides this, we also could identify ubiquitination sites in a number of other viral proteins, suggesting that the ubiquitination system is quite active in modifying these viral proteins. We yet, of course, don't know what the activity would rely to, so whether this is pro or antiviral in this case, but at least it would suggest that the ubiquitination system is quite heavily involved in the biology of SARS-coronavirus replication. It's not only the viral proteins, but also their binding partners that are in part regulated. There is, for instance, regulation of ACE2. We could find a novel ubiquitination site at position 625. And in general, induction of these ubiquitination sites over time. This may suggest some regulation of ACE2 expression over time, besides its known shedding by metalloproteinases. So it could be that there's a number of regulations that could be in place to regulate ACE2 levels and thereby the entry port of SARS-CoV-2 during virus infections. And ACE2 was not a unique example here. If you now look at all the phosphorylated and ubiquitinated proteins and ask whether they are binding to individual viral proteins, we could identify 180 proteins that were actually present in the interactome, and that showed post-translational modifications after virus infection. And this experiment actually prompted us to ask whether these interactions that we identify by this interaction proteomics may relate to some downstream activity that we should be able to map. And in order to study that, we used a so-called network diffusion approach, whereby we went back to the individual ORF expressing cells, and we asked whether the changes in protein expression that we observe after introducing these viral ORFs into these cells may somehow be functionally linked to their interactions that we also identify in the same cell types. And as a basis to link these different parts, we use the reactome network, which is a network that connects functionally active proteins in given networks. So basically, we then came up with subnetworks of binding proteins that were partially linked with other proteins to proteins that are changing their expression profile. Either these proteins were upregulated or downregulated after expression of this individual viral open healing frame. And what you get then is subnetworks of proteins that are binding to a given open reading frame. In this case, for instance, ORF7b, to proteins that are changing the expression pattern after expression of the same open reading frame. And the networks that we identified here are not caused randomly. We can find by permuting this network that this is a highly significant network that is formed, and we use this network then to identify the activity of these individual viral open reading frames in order to understand what the activity would be. And if you on top of this, we could, well, and then you basically get small subnetworks that look a bit like that. So this is, for instance, ORF3 or SARS-CoV-2 that would associate to cellular proteins here in gray. And these cellular proteins are functionally linked through the reactome pathway to downstream proteins that are changing in expression after expression of ORF3. In this case, for instance, ORF3 was enriching for proteins that are linked to autophagy. So we find, for instance, proteins that are related to autophagy induction, and we also find proteins that are known to be upregulated during autophagy induction. And we could not only do this for ORF3, but also for many other ORFs, such as, for instance, ORF7b, that we identify as a protein that is regulating innate immune responses by binding, for instance, to the mass, and through regulation of downstream candidates such as NF-κB and others. Also, we find a functional relation of ORF8 to the TGF-beta pathway and corresponding downstream protein expression patterns that would solidify the functional activity of this ORF and would link this to these interactions we identified. So we got now these little subnetworks, and we enriched these subnetworks then with the additional data we had from the same cell type. This is based on the protein expression, the ubiquitin, and the post-translational modifications. And that actually allowed us to identify pathways and to find their regulation in an unprecedented manner. And I just want to show a few examples here. This is, for instance, the autophagy pathway, which we identified to be targeted by ORF3. ORF3 is, for instance, finding the Hobbs complex, which is known to activate the autophagy pathway. This interaction has also been identified in neurochromats data, actually. And we also identified other candidates of the autophagy pathway that are associated to ORF3 or to NSP6. And in addition to these interactions, we also found proteins that are differentially phosphorylated after SARS-CoV-2 infection. And here you can see in bold borders proteins or regulatory sites of proteins that are known to be associated with autophagy induction. Besides these regulatory sites that are in part well studied, we also found other co-occurring sites that are also upregulated during virus infections, and we assume that there may be some common or some activity of these phosphorylation sites, although they are not very well studied at this stage. This autophagy induction could also be mimicked actually by simply overexpressing of 3A. You can see here, for instance, expression of LC3B after ORF3 activation, transduction of using SARS-CoV-2 ORF3, while the corresponding ORF3 of SARS-coronavirus was not doing this. So clearly there's some specificity here to regulate the system, and it's not only the single ORF that is doing this, but also we can see this upregulation of autophagy pathways after SARS-CoV-2 infection, suggesting that these networks are really functionally linked. Besides autophagy, we also found a quite dramatic regulation of the innate immune response. We find a number of proteins, such as, for instance, ORF7b that I mentioned before, to target central regulators of the innate immune response, such as MAFs and UNC93B1, which is involved in TORC receptor signaling. But we also identified other targets of the virus, such as CHAC1 and CHAC2, which are involved in cytokine interferon signaling, suggesting that this virus has many different ways to target the innate immune system in parallel in order to take out central innate immune pathways in the course of virus infections. Lastly, we also identified regulations on the TGF-beta pathway. So here, for instance, I already alluded on the association of ORF8 to the TGF-beta complex, but we could also identify downstream signals of the TGF-beta pathway being modulated, such as, for instance, differential phosphorylation of MAP kinase proteins, or also differentially regulation of integrin signaling here. So clearly, to summarize this, we think that the interactome of these viral ORF proteins reveals many novel interactions, some of which are very specific to SARS-CoV-2, some of which are shared and may be explaining why these two coronaviruses are of particular pathogenicity. Our data allows to link the activity of single viral ORF expression to downstream protein expression patterns, and thereby gives a functional link between the activity of the interaction to some downstream events. And you could also map post-translational modifications on regulatory circuits that are differently activated after virus infections. But the question is, of course, how can this help to fight or to gain knowledge and to fight COVID-19? And here, basically, we considered all these different data sets, and we basically asked whether any of the proteins that and pathways that we identify in this omics approach would also be represented in the activity of drugs that are, for instance, deposited in drug bank or Camber. Therefore, we mined these databases and identified drugs that would be, for instance, targeting the pathways that are regulated by SARS-CoV-2 or by the individual proteins. And we functionally studied these proteins using a light microscopy-based screening platform that basically allows to test for the expression of a GFP-expressing virus that we kindly got from Volker Thiel. And this pathway, this platform basically allows us, for instance, to look at replication of the virus in a time-dependent manner. And you can see here, for instance, an increase of the GFP signal in mock-treated cells, while, for instance, interferon-treated cells are very much impaired in the GFP expression, suggesting that this is a very active antiviral compound that would inhibit the virus. And this is also in line with quite a bit of literature that came out recently on the activity of type 1 interferons against SARS-CoV-2. So altogether, by now we tested around 70 drugs, some of which are shown here in a combined manner. In the y-axis, you can see the GFP intensity that change that we can see after treatment with these drugs. Here, for instance, we can identify a number of candidates, such as, for instance, metalloproteinase inhibitors, pneumonostat and marinostat, which are actively inhibiting the virus, as well as interferon alpha and interferon gamma in this system. But we also identified kinase inhibitors, such as, for instance, gilteritinib or ibatacitinib, which is an AKT inhibitor, inhibiting the virus. We are particularly excited by ibatacitinib because this was actually one of the kinase inhibitors that, it is inhibiting AKT kinases. And as I told you before, we identified AKT as being one of the phosphatases or kinases that are phosphorylating sites, potentially phosphorylating sites on the the nucleocapsid protein. And it could well be that this is basically directly affecting the activity of this viral protein. Other kinase inhibitors, interestingly, have a pro-viral effect, and these are particularly BRAF inhibitors. At least in this system, we can clearly see that inhibiting this kinase would increase virus growth. And there's also other drugs, such as, for instance, DNA damage inducers. They're often used in cancer treatment that are also inhibiting virus replication in a quite effectively. So to summarize this, we think that this is a very valuable resource for further analysis. So all this data is actually provided online right now in a preprint server and available in Intact and Pride. The data highlights proteins and pathways that can be targeted by drugs. And we feel that together with other data sets that have been published by other groups, this data set is highly valuable to identify the activity of single proteins and also to understand better how this virus is interacting with human cells and how this would affect cell viability and our human viability, basically. Finally, I want to thank people who have been involved here, particularly members of my lab. The ones that contributed here are shown in bold, so it was quite an interesting, intense effort in the last couple of months. Here, the peak of Germany is the highest peak in the German Alps. But in particular, I also want to thank our collaborators, Matthias Mann at the Max Planck Institute of Biochemistry, and Özge particularly for doing a lot of proteomics here as well. We also have other essential collaborators, Bernhard Küster on kinase inhibitors and considering many phosphorylation events that we found, as well as the people who are more working on transcriptomics. And people who contributed providing essential reagents for coronavirus research, such as John Zibur, Volker Thiel, and Roman Wölfer. And we are very happy for that. And again, I want to thank you for the opportunity to present our work here. We feel that we would be very happy if you used it and it becomes useful for everybody involved here.
Great, thanks. That was a great talk. You actually answered a lot of the questions that the group was having as you were talking. So we'll just come up with a couple of the ones that have come in on the Slack, although I think you also answered them while you were talking. But one comment perhaps that you can say is that one person's asked, they've said that the N protein is conserved among coronaviruses and they don't think, therefore, that it would be a good target for SARS-CoV-2. What are your thoughts on that?
You mean a specific target for SARS-CoV-2? I mean, in fact, well, so what we identify here are motifs that are differentially phosphorylated, for instance, here. And we don't know exactly whether, for instance, the kinase inhibitor that we're using is acting on the N protein itself or on any other cellular protein that would, for instance, be important for SARS-CoV-2 activity. I think that using, of course, this would be something that would need to be evaluated, whether it is now acting on the N protein itself or whether it's something else. that is affected by these kinase inhibitors. But in principle, whatever would inhibit the virus would be beneficial at this stage, I believe.
Great, because you've already answered the next bit with what you gave in your talk, which was, did you identify any peptides corresponding to the S protein? But you obviously showed your ubiquitinated peptides from the S protein, so I think that part of the question is already addressed. But when we have another one that says, did any of the already tested drugs that are obviously a bit more in the media, like remdesivir or Trump's favorite hydrochloroquine, come up in your search as a potential target, or did they not?
You know, remdesivir is a nucleoside analog that, well, of course, in some way you may be able to link it to transcriptional activities, but it would not be something that would come up here. We tested remdesivir. It's a fantastic drug. I think I'm a big fan of it. It's inhibiting at least the virus in this system. For chloroquine, we also see effects on the virus. Chloroquine in our hands is also toxic, particularly at higher concentrations. So anybody conscious should not really swallow a lot of it, I think. But but it has, of course, an inhibitory effect on virus infection. If you would, for instance, see the autophagy pathway or endosomal pathways as a target, you would also come up with chloroquine, because it is a compound that would inhibit these particular events. Of course, any of the drugs that we are testing here would require additional testing. So this is just an in vitro assay. And if there would be any, I would not advise anybody to swallow that without any further testing. But at least we come up with candidates that is based on the activity of the virus and that is based on knowledge on the pathogen in order to identify something that would, for instance, allow to target the virus more specifically than a broad antiviral compound.
Great. And have you tried to cross-reference your data with any of the clinical plasma proteomics data to see if any of the regulated pathways actually pop up in those samples as well that match your?
We did not do that, but it's a good point. I will note it down.
And another question's just come in. Did you see any other interactions of the spike protein other than with the ACE2 receptor for either virus-like, and then they've written something else which I don't really understand, BSG. Maybe that's something for a virologist.
Yeah, I think we found an interaction of basogene with spike protein. I can't recall specifically the interactions of spike, but all of them are actually listed and provided in the preprint publication. So there is a number of interactions of spike, of course, that are specific, that we can identify. Some of them are also being identified by other people, of course. So I think the entire data set itself appears to be quite congruent to others. It extends to some extent. It's as any interactome, I think one has to see them as entities that are enriching themselves or each other. So yes, we find quite a number of interactors that are actually known, and a few novel ones as well, of course.
Great. And if you had to pick one of the drugs that you think should go further into trials, or that you might consider, if you, worst case scenario, got the virus yourself, what would you take, or what would you recommend? When
that's a disclaimer I just had. You know, I would. I think it would require further testing.
Sure.
And we are, of course, trying to do so in more sophisticated models. Please keep in mind that this is a cell line model that is based on A509 cells. It may well be that in a more complex systems, such as RE cultures at least, the responses may be slightly different. This is, we are currently testing this in more sophisticated models, and then we will try to promote them further. We are, what is quite interesting is that some of the drugs that we tested had been used for clinical trials before, for instance, the metalloproteinase inhibitors that just got off the clinical trial because of long-term effects. But I could well imagine that short-term treatment with these kind of compounds may be less detrimental or less affecting the long-term effects that are known for this type of compounds. And yet we see very promising effects for this type of drugs. We think, at least, what I should say is that I'm a big believer that combinations of different drugs would be very beneficial. So it's not a single activity, but that there's different activities that may be targeted. And I think this is something that may be explored in the future a bit.
Great. So thanks very much. I think that's it for all the questions that have come in so far. So to the attendees, as mentioned earlier, we'll have a questionnaire to get feedback on the meeting and a form requiring certificates of attendance. You'll be able to scan a QR code shortly. Like to say a huge thanks again to both of our speakers and to everyone for coming along, and to our sponsors and those committee members who have been working away in the background to make this webinar possible. So yeah, thanks everyone, and have a great weekend, and we'll see you in 2 weeks.
Thanks.